Microplastics are becoming a global concern due to the potential risk to human health. This study focused on widely used polypropylene-based baby feeding bottles to develop a cost-effective protocol suitable for MP study of many plastic products. This protocol provides the details of sample preparation, identification, and characterization. It can substantially benefit future studies of microplastics released from plastic products. Microplastics released during formula preparation. Soak the baby feeding bottle in 95 degrees Celsius deionized water to sterilize the bottle. To avoid the bottle floating, slightly press it using a stainless steel tweezer to ensure the whole bottle body is immersed in the water. After 5 minutes, take the bottle out and move it to a clean glass disc to wait for air drying. After air drying, pour 180 milliliters of 70 degrees Celsius deionized water into the bottle. Then cover the bottle immediately using a glass Petri dish and place it onto a shaking bed. To stimulate the formula mixing process, shake the bottle at a speed of 180 RPM for 60 seconds. After shaking, move the bottle to a clean glass plate and allow it to cool down. Sample preparation for microplastics identification and quantification. Membrane filter is critical for microplastics capture, visualization, and detection. We used the gold-coated polycarbonate membrane filter to capture microplastics. Place a piece of membrane filter in